Hello and good morning, everyone. My name is Maria DeRosa. I'm the Dean of Science here at Carleton University, and I'm really delighted to be able to moderate today's New Faces at Carleton panel for Data Day. Thanks, everyone, for joining us. So just a quick overview. The goal of our session today is to provide an opportunity to hear from some of our newest hires in data science about their research and their thoughts around the future of this exciting and impactful field. Carleton, you've heard this already from the opening remarks, but Carleton is building some really great broad-based interdisciplinary expertise in data science coming from all corners of our university. And I think that's going to become apparent today as we listen to these esteemed panelists and hear all about their work. Just a little overview of the plan. After a quick introduction of our panel, each panelist will have about 10 minutes to walk us through their research, and they'll be sharing slides and you'll have a chance to ask questions. So if there are any questions that come up during the presentation, feel free to pop them into the Q&A and I'll be moderating that. We'll have time for a couple of questions after each presentation, and then hopefully more time for a more wholesome discussion towards the end of our time together. All right, so with that, let me introduce some of our panel members. First up, we're going to have Professor Alan Sung from the School of Computer Science here at Carleton. He did his PhD at the University of Waterloo and a postdoctoral fellowship at the National University of Singapore before joining us in 2020. Welcome, Alan. We also have Professor Carlos Rasa from Systems and Computer Engineering here at Carleton. After a PhD at the Sorbonne and PDF at University of Alberta, he took a faculty position at Ontario Tech before he joined us just in July 2021 as an associate professor in systems and computer engineering. Welcome, Carlos. We also have Professor Sanjina Dang from the School of Mathematics and Statistics. After a PhD and a postdoc at Guelph, she actually took a faculty position at Guelph and eventually at Binghamton University before joining us in 2021 as a Canada Research Chair in Data Science and Analytics. Welcome, Sanjina. And last but not least, we have Corinne Millard from Geography and Environmental Studies, actually a Carleton alum, which is a wonderful thing, and was a research scientist in Environment and Climate Change Canada before joining us in 2020. Welcome, Corinne. Great. So with that, let's get started. And I'm going to ask Alan to take the floor for us and give us a little overview of his research program.
Okay, thank you for the fantastic introduction. Let me get the slide share on. So hopefully, there we go. So hopefully that should work. All right, so let's suppose you and your close 4 friends are trying to pick a movie to watch, right? And if you're trying to pick a movie, then you need to, well, you all have different preferences. You all like different things. So let's vote on it, right? So here are you and your 4 friends. There's 5 of you. You voted on these various movies. And so let's say Iron Man's the winner. But you think to yourself, I don't really like Tony Stark. I really like Star Trek. But if I can't get Star Trek, maybe I can get Star Wars instead. And so, wait, I changed my mind, right? I changed my mind. Instead of voting for Star Trek, I now vote for Star Wars. And let's say by the magic of tie breaking, the winner is now Star Wars. This is what we call strategic voting. You've, in some sense, ceded your favorite choice, the thing you really wanted, in exchange for something to prevent something you don't want to happen. And so this is called strategic voting. But wait, other people can do this too. So here, the yellow player, the yellow agent, has said, wait, I also want to change my mind. I really don't like Star Trek or Star Wars, but I'll vote for the Captain America instead. And this is now called iterative voting, where agents can change the vote in turn. So why do agents vote, right? What is happening in this situation? Why do agents choose to vote at all? Well, there are a number of reasons why agents would vote in an election or in a smaller stakes affair like picking a movie or picking which restaurant to go to. You might be voting as a way of expression, and that's very important, but not our focus today. Instead, our focus today will be looking at voting as a way to change something, right? This idea is that the only reason you want to cast your ballot is that just in case you're the pivotal voter, right? You have some small chance of altering the outcome of the vote, and therefore become the pivotal voter. And so the chance of you being able to alter the outcome is what motivates you to vote, or one important motivation for you to vote. And using this, we can now view strategic voting as a way of as observing a chance to change from your favorite vote to get your second choice, or at least to prevent a bad outcome from occurring. And so this is the model we'll be looking at today. And what I did was that I took this model of voting, of iterative voting, and applied it to a social network. Whereas iterative voting makes sense in a scenario where it's you and your close four friends. I want to try to use this model to extend it to larger populations. Then it doesn't make sense that, for example, you can see everybody's votes, right? That you know exactly what is happening, and you can jump in when you see an opportunity to perform strategic voting. And so to generalize this to a network, well, we need to make it harder to see everybody's votes. For example, we might say that you might be here in the social network, and you have some friends on this social network. This social network is very large. It might have thousands or millions of nodes. Millions of individuals, but you're only able to observe information from your friends in your social network. And so this changes the idea of what it means to vote strategically. Instead of observing an opportunity, yes, I can break ties the other way now. I'm now the pivotal voter. Instead, now we have to work with pivotal probabilities, where we have to work with stats, right? There's a lot of math involved here, but we need to estimate what is the likelihood that we can actually affect the outcome of a particular vote. And so this is a study that I carried out, and it's based on simulations, because we'd like to be able to work with real world data. But you can imagine what that real world data would look like. It's a social network data, not only with individuals identified in the network and their friends and family, but also their political affiliation, what they voted for, and that just seems utterly impractical to obtain this data. And so instead, we use simulations. We simulated social networks using standard random graph models. And I'll show you a bit of the results. We should just jump over the methodology. This becomes very technical. In fact, this visualization is a bit hard to read. But in effect, we're looking at the blue line here. And this blue line indicates how much happier agents are having voted strategically rather than truthfully, based on what the elected outcome would be. And so the higher, the better. And what we're observing is that agents that vote strategically generally are happier with their outcome rather than having a truthful vote based on their non-strategic preferences. And the orange line here also looks at how good is the strategic outcome? Is it better? Well, it's always better than having voted truthfully. Better in the sense that everyone in the election, on average, is happier with that result. And in fact, what we do see is that when we vote strategically in this kind of scenario, we get close to the optimal outcome. The optimal outcome is obtained when we can read everybody's minds and just pick the best one for everyone, right? Pick the best outcome for everyone. One of the other things we also studied in this piece of research was homophily, right? This idea that your social network is generated in a way that's biased. And in fact, people tend to connect with people who are similar to themselves. Picture the last 10 people you spoke to on your Facebook or Instagram or WhatsApp or WeChat. Right? These people are people who have similar backgrounds, similar interests, similar socioeconomic status, similar cultures as you. Right? People who when you make connection with someone, you typically make connections with someone who is similar to yourself. And that alters the way that the social network structure looks. And in fact, what we did do is that we modified the network generation protocols to incorporate homophily explicitly, so we could generate more realistic social networks. And what happens when we look at these networks with increasing amount of homophily is that homophily actually reduces how frequently people vote strategically. And in fact, agents would vote their top preference more often. That's what this in fact does. And as a result, people are just less happy with the voting outcomes, right? Averaged over the entire population, not just people who have sort of not altered their votes. Everybody is just more sad with what you end up picking as a result of that election. And so why does this happen? Well, what happens is, let's say you're this agent down here, and this is what you observe in your social network, right? You have a very large social network, and amongst your friends is a race between well, orange and greens, right? And you think, okay, well, I like green better than orange, so I'll just vote green here. But what is actually happening is, because there's homophily in this network, you are preferentially connected to people who think similarly to yourself. And so here, the actual race is between the orange and the blues, and the greens have a very small slice of the pie. And so your vote having gone to the greens in this particular case, it's not a vote that's effective. Right? And so by having this sort of homophily effect in the network, if you happen to be an agent whose belief is sort of very in the vast minority, you might not know that, because you think your belief might be more commonplace in the population than it actually is. And so this gives you a taste of what's called computational social choice, right? This idea of studying social choice, which is how do communities choose something together as a whole, and using computational tools to tackle this problem. And it is a branch of multi-agent systems. And more generally, multi-agent systems is concerned with modeling communities of decision-making agents, where the agents not only make decisions, but they know other agents are also reasoning about the situation and making decisions themselves. This is a cartoon I like to use in class to sort of discuss this. So we think of classic AI as the study of rational and intelligent systems. So here we have a robot. The robot really likes this blue box at the bottom, and there are these other boxes in the way. It's a classic AI problem in like the 70s, right? It figures out how to move these other boxes, and okay, great. It now has the blue box. This is a classic AI problem. There's a single rational intelligent system at work. What happens when there are other robots? Right? This is what multi-agent systems is concerned with. Not only are you making decisions in this environment according to your own preferences, but you also understand that there are these other agents that are in this environment who have preferences. You might or might not know what they are, but they're also reasoning about this problem. And now you're thinking, well, what do I do now if I was this robot? Do I grab these blocks? Are the other robots going to run away with my block? Right? And so this gives you a different lens for thinking about these kinds of problems. Generally, we think about in multi-agent systems, how do rational agents behave in particular systems that we observe? And how do we design a system to induce certain behaviors that we find desirable in rational agents? For example, how do we allocate resources? How do we do so fairly? What does fair even mean in a mathematical or computational sense? And I've placed these asterisks over rational because I think that's a bit of an easy assumption to make, that, okay, we're only concerned with very logical agents, when that's not necessarily true, because a lot of these models extend to incorporate what are called behavioral components, right? That people might not, or these agents might not make decisions fully rationally. And so I'm just going to run through a couple of more recent work. This is work that I've done with several researchers in the US, looking at fair allocation of health resources to homeless youth populations. And currently we're using multi-agent systems models to simulate COVID interventions and how different lockdown procedures and vaccination procedures might affect that on a social network basis. This is a very exciting project where we're looking at modeling using Schelling's segregation model, where we have this sort of grid world, and we want to look at how do minorities choose when they might assimilate into a majority culture, and how does that affect intergenerational wealth. And my current grad students are working on projects such as looking at referral bonuses and how does altruism play a role in that, looking at how AI might play social deduction games, and evolving autonomous car strategies. Okay, thank you very much.
Fantastic. Really, really clearly explained. So if anyone has any questions, be sure to use the Q&A to pop your questions in, and I'll just ask a quick question, Alan. You mentioned the challenge in gathering data related to these sorts of things. You say you have to model them, and because of the challenge with getting this sort of data. So how does that impact the kind of work that you're able to do? You know, for given the challenges around accessing these sorts of data?
Yeah, so a lot of my research in the past have been simulation based, because getting a hold of this data is tough, and frequently it's locked behind closed doors, right? Like Facebook has this data, no doubt.
Exactly.
Or Meta has this data, and that's difficult to get access to. And so simulation is an easy approach to take, because it circumvents a lot of the challenges and also ethical issues of having this data in the first place. But where possible, I think it is nice to be able to use real world data. That's really good, right? And in the health interventions, we use very sensitive health data that was gathered on the ground for homeless youths in LA. And that was a very, very nice opportunity to be able to demonstrate that.
Wonderful, thank you. Well, I think what we'll do is, in the interest of time, we'll move on to the next speaker, but definitely keep an eye, Alan, on the Q&A. If questions pop up, you should be able to interact virtually through the platform to answer questions, and we'll also direct any questions to Alan after the session if needed. Thank you so much for that. That was great.
Thank you.
So next up, Dr. Rossa, would you like to take the stage and share a little bit about your research?
Sure. Thank you very much. Let me just share my screen here. Just 1 second. Okay. Hope this is visible. Yeah, so I'm going to talk about my research. I said I joined Carleton in 2021 after 4 years at Ontario Tech University. And my research is mostly focused on surgical robotics. So I selected a few projects, ongoing projects here, that I think are relevant to the discussions today. But before I go into them, just a brief overview of the areas of research in my lab. So there are 3 main areas: robotic surgery, haptics, and instrumentation. If you go explore the overlap between robotic surgery and haptics, this leads to a lot of interesting research problems here in telesurgery, collaborative human-robot surgery, and the development of precise medical devices to improve the outcomes of surgery. In the overlap here between instrumentation and haptics, we have force feedback. So if you're using a console to operate or to control a robot that operates on a patient, we want to recreate the sensation of touching the tissue, even though we are not directly touching that tissue. So that's where force feedback comes in. We can implement methods to give the surgeon that is using joysticks the sensation of touching the tissue as if they were operating directly on the patient, even though they are doing that through a robot. The last overlap here would be between robotic surgery and instrumentation, and we see image-guided surgery here. So how do we instrument these robots or these surgical tools to either provide more information about what is happening, what is being manipulated in real time, or inform the robot how to perform the surgery autonomously or under human supervision. So here we can explore how the robot and all these sensors can work together, and also develop new diagnostic and imaging tools. And the research in my lab is at the center of these 3 topics. So I've selected a few examples here of ongoing projects that I'm going to talk about, just to give you a better overview of what is going on in my lab. So the first topic, just a really high-level overview of percutaneous nephrolithotomy. This is a procedure to remove kidney stones from a large kidney stone. So it's a surgery that involves inserting a tool through the patient's back towards the kidney, and then reaching for the kidney stones and then breaking them down and take them out. Even though this is a fairly common procedure, it's very complicated to learn, and the failure rate is quite high. So I've been working with this company from Toronto. They developed a simulator to help these surgeons just develop their skills in a virtual environment scenario. And they basically can do a full simulation of the procedure before they go into the operating room. And we can also display different scenarios to them and see how they perform. So after collecting a lot of data from these surgeons, we realized that this data is quite useful. We have the trajectory of the tool, we have how much force they apply to the tissue, we have how much time they took, the acceleration, all this information is available to us. And the question then was, what do we do with all this information? Can we perhaps use this to either teach novices how to perform the surgery, or even teach a robot how to help a surgeon perform the surgery in the operating room? So that's the goal. So based on all this data, we created two main things. The first one is a way to assess how inexperienced surgeons do the surgery in the simulator and provide the cues to help them improve. And the second one was to use all this data to teach a robot how to perform the surgery either autonomously or in collaboration with a human operator. And that's the figure you see here at the bottom. This is the experimental setup in the lab that used the data from the simulator to teach the robot how to go from a given point to the kidney stones, either autonomously or under human supervision. In that case, then we have a joystick on the left here that the human operator uses to control the robot. And in this process, we have many different levels of autonomy that can be granted to the robot. For example, the robot can control, make sure that the tool always passes through the same entry point in the tissue, whereas the human operator controls the position of the tool tip. Another option is that we have an optimal trajectory that you can determine from the simulator, from the data we got from the simulator, and then we can control the robot to kind of be attracted towards that path. Even though it doesn't have fully autonomy, you will feel the forces attracting you towards that optimal path as you perform the surgery using the console. So this leads to so here's the robot in action in that scenario. You see the robot controlling the insertion point in the entry point in the tissue that is fixed, and the operator then controls the position of the tooltip. This leads to an interesting mathematical problem. Here is just a simple equation that is used to calculate the speed of the robot's joints. And it has this interesting matrix in it, and that's the one element of that matrix. There are, I believe, 64 of those. So it's an interesting mathematical problem to see in action and see it working during surgery. So that's one project, and we are still working on it in collaboration with the company. And this led to several other projects. Here's one of them. We realized that these commercial devices that we use to control the robots are not very reliable and not very stable most of the time. So we decided to develop our own, and this is the result of that is a haptic device that it can be used for surgery. Only has brakes, so it has never go unstable. And for this type of procedures, we want something that really cannot harm the patient or cannot or has minimal chance of failing during the procedure. So moving on to another project here is the idea is to instrument surgical tools to provide additional information about what is happening when a robot performs the surgery. So here is a simple example. We instrumented surgical needles that can be used in conjunction with this robot. And these surgical needles are able to determine the electrical characteristics of the tissue at the tip. So we get a database now from several different tissues that has the impedance signature of the tissue to a given range of frequencies. And based on that database, when the robot performs the surgery, we can do some sort of real-time classification method to determine what type of tissue the tool is going through at a given time. And based on that, either update the robot operating strategy or simply display that information to the operator in real time. So we validated this with a number of different tissues and in conjunction with the idea of instrumenting a robot to do that. So here we have the tool, instrumented tool attached to the robot. The operator is controlling it, and now we make the forces the operator feel at the joystick level proportional to what the robot is measuring at the tip of that tool. So that allows you to precisely place the tool in a given target with an accuracy here that can range 5 micrometers, which would be impossible to do manually. So an extension of this came up in collaboration with the Ottawa Hospital. If you're able to identify the tissue at the needle tip, maybe you can use this, for example, in tissue biopsy. And the procedure we decided to target was prostate biopsy. So when a patient goes for prostate biopsy, they first get an MRI of the prostate, and then the doctor will look for inclusions in the MRI image. And if anything is detected, any lesions present, then they go for biopsy. But the biopsy is done under ultrasound imaging rather than MRI. The problem, as you can see here, is these images are quite different. We get an MRI telling us where these inclusions are. And then during the biopsy, all we see is the ultrasound image has a lower resolution. And the objective now is to insert a needle into these lesions and sample them. So we have a rough estimate of where these needles should go, but we can't really be sure. There are, of course, methods to register these two sets of images and find the equivalent location of the lesion in the ultrasound image. So the surgeon will try to target that, but it doesn't work all the time. And if it doesn't work all the time, that's a problem, because if you get a negative biopsy result, we don't know if now that a negative result is due to the absence of cancer or simply because the target was missed or the image registration didn't work. So typically the patient would go for a second round of biopsy. The idea then that came up with the Ottawa Hospital was to use the sensorized needles to determine whether or not the tool is placed at the specific location where the lesion is before taking the sample. So we use the sensorized tools to determine if it is appropriately placed, and if it is, then you take a sample, and if not, you just reposition the tool until we reach that lesion. And the lesion and the tissue themselves will have different characteristics in terms of electrical properties, so we can determine where exactly this tool is. And we are currently working on this with the Ottawa Hospital and a group of urologists from there. Another extension of this instrumentation work was to now, instead of just using one needle and measuring what is happening at the tip of that needle, what if we use several needles at the same time, which is the case for certain types of procedures, such as brachytherapy of breast and prostate cancer. We may have up to 20 of these needles inserted at once in the tissue. So instead of looking at the tip, what if we look at the impedance across these different needles? And by doing that, we can now move towards a map of the conductivity in the tissue and see if the lesions show up in that image. And that's the preliminary result we see here. It's not very good at this point, but we can clearly see that we can identify inclusions in the tissue by simply inputting these needles in the tissue and determine, based on the data we collected previously, what that tissue is. If it is cancerous, if it's not, it's just healthy tissue, an inflammation or something like that. So as was previously mentioned, I just moved here from Ontario Tech, so I was fortunate enough to be able to bring all my equipment from there. So here is just some of it. I'm always looking for collaborators, and if anybody has use for this at Carleton, I'm more than happy to collaborate. Ultrasound machines for research, robot arms for surgery, COVID lung ultrasound simulators, and surgical electromagnetic tracking system, and of course 3D printers, as most of the labs we have. So that's it for my brief talk. If you have any questions, feel free to email me, and my address is here. My office in the Canal building. Everybody's welcome to stop by, take a look at the lab and the infrastructure we have there. Thank you for listening.
Fantastic. So there is actually a really great question that's popped up from the audience, from Professor Roby. It's interesting that our bodies are somehow different from one person to another. I wonder whether this affects the model trained on training data of a selected population of patients. Can we claim that this training sample sufficiently represents a general population of patients?
Yeah, that's a very, very interesting question. We are always looking for different samples, for a variety of samples. The more samples we get, the more accurate the model gets. We tried to use this with in a vet clinic, after they removed some specific tissues from dogs, we are able to identify them. But we need a relatively big sample in order for this to be accurate, because there are different stages of the disease, and sometimes if you just use one stage when you are in a different stage, that may not work. Yeah, that's definitely the critical point is a good database for
Makes sense
training.
for all these issues. No, that's great. Thank you so much. If there are any other questions, feel free to pop them into the Q&A, and we'll take some time at the end to address them. But thank you very much, Dr. Rossa, for that presentation. And next up, we'll have Dr. Sanjina Dang take the stage and share her research with us.
Well, thank you, Maria. Let me just share my screen. Well, first of all, thank you, Maria, for the wonderful introduction. And thank you, everyone, for actually being here for the presentation today. Sorry, I think it may have closed my screen. All right. So my primary area of research is cluster analysis with applications in bioinformatics. So just looking at it from a data science perspective, we can think of data science as essentially using statistical/mathematical models along with computational tool to work with interesting problem that arises in a variety of different domains. So when you think about bioinformatics, we are essentially using statistical/mathematical models along with these computational tools to tackle interesting problems that arises in bioinformatics, in biological sciences. So in that sense, you can think of bioinformatics as a specialized data science. So what is cluster analysis? So cluster analysis assigns data points into different clusters or groups so that the observation in one cluster is as similar, observations in a cluster are as similar as possible, and observation in different clusters are as dissimilar as possible. So let me start by talking about what exactly is clustering. So here is a toy example on my left. As you can see, we can clearly, looking at this toy data set, we can see that there is a cluster structure in the data. So we have one cluster on the top right-hand side and another cluster on the bottom right-hand side. What we are really interested in doing is developing these models that can classify these observations into different clusters without knowing or without having any prior information on the cluster structure of the data. So let me show you a quick small animation. So I started the algorithm by randomly assigning the observation into two different clusters. And I'm letting my algorithm run and for it to be able to detect the underlying cluster structure in the data. So my work primarily focuses on model-based clustering. So in model-based clustering, we are not just interested in assigning the observation into different clusters and just interested in identifying what the cluster center looks like. What we are interested in is using a statistical model to better understand the shape of the data and to be able to build a model that can capture the underlying data generating mechanism as well. And in the meantime, we also get a probabilistic interpretation of the cluster as well, because an observation that is at the borderline could actually have a fairly high probability of being into two different clusters. So for a two-dimensional data set like this, it's easy to visualize. And by looking at the data alone, we can actually see that there are two cluster structures. But what becomes challenging is when you're dealing with high-dimensional data. So high-dimensional data, visualizing them, it's not easy. And clustering itself becomes even more challenging. So cluster analysis is used in a variety of different fields. I'm going to talk about some examples where cluster analysis have been widely used. One of them being image segmentation. So essentially trying to find regions or segments in an image based on some similarity measures. Where would this image segmentation be useful? When it's used in self-driving cars. It's also used in classifying medical images to identify disease versus healthy regions in the image as well. Another example of where cluster analysis has been used is to detect anomalies/fraud detection and for using them to actually minimize the risk. So Lee et al. In 2015 utilized cluster analysis to detect abnormal flights. Another example of cluster analysis, where cluster analysis has been used, is for market segmentation. So in this example, suppose you have data sets from consumers about different consumer report on fashion trends. Let's say we clustered the data and you have three different groups here. One group that is driven by fashion, another group that is interested in fashion, and the third group who actually purchases only when necessary. So why would it be useful to identify these groups? Let's say you were developing a new product. You can actually now, once you know these underlying similarities or groups in the data, now you can actually develop targeted or tailored ads or tailored advertisements to these groups so that you can maximize your profit. And in some cases, you may only be interested in targeting a particular subgroup as well, rather than looking at the entire population. Other applications, so cluster analysis is also used in search engines to increase retrieval precisions. It's also used to detect clusters or communities in social network. So what my work primarily focuses on is clustering various different types of biological data sets. So my current research focuses on developing clustering models for gene expression data, microbiome data, as well as looking at integrative clustering platforms where we can utilize information from multiple different biological data generating platforms. And one thing I've been very lucky is, I've had the opportunity to collaborate with researchers from a variety of different fields. I've had collaborative opportunity in structural biology, metabolomics, nutrigenomics, genetic data. So working on all these fields, I have actually gained a good idea of what kind of statistical tools are actually needed in order to be able to meaningfully integrate information from various different platforms to gain a good underlying unifying view of a particular problem. So as I mentioned, some of my work focuses on gene expression data. So what is a gene expression data? So in gene expression studies, we are essentially trying to quantify the expression levels of thousands of genes. And two platforms are primarily used, microarray and RNA-seq. So we have developed various different statistical models that can cluster these microbiome data and RNA-seq data so that the genes that have similar expression profiles are clustered together. And again, microarray data, RNA-seq data, they use a completely different they use a completely different way of generating the data. So the underlying statistical models are very different, but the goal is to be able to cluster the data so that we can understand which genes behave similarly under different conditions. So here's an example of one of our recent work where we clustered an RNA-seq data. So on the image on the left, as you can see, we have expression profiles of different genes, and this is before clustering the data. And as you can see, it's fairly messy to be able to identify which genes behave similarly. We run our clustering algorithm on the data, and on the right is what you see are the 8 clusters of genes that we got. And as you can see, the observation in a cluster are much more similar to each other, and observation in different clusters have a different trend. So essentially, what we are doing is we are developing these statistical models that can identify these, that can actually detect these clusters automatically without us telling these genes should be together and these genes should be different. And of course, it is possible to incorporate if you have any prior information. You can actually incorporate those information as well. But this clustering, the one that I'm showing here for illustration, is essentially we are not using any information. We are letting the algorithm detect the clusters. Another area of my research is on developing clustering models for microbiome data. So in our bodies home to a diverse and complex microbial community, which are collectively known as microbiota. So these are bacteria, fungi, viruses, archaea, and so on. And it's very important to understand how various different biological and environmental factors, such as diet, your age, your environment that you're living in, as well as exposure to antibiotics, all these factors affect the microbiome composition that you have. And varying composition of these microbiome has been known to be associated with several different diseases. So here is an example from one of our recently submitted manuscripts. So again, when you're looking at these data sets, these are massive data sets. So we apply clustering, our novel clustering approaches on this data set. And because we want to visualize this massive data set, so what we do is we actually project this data set into a two-dimensional space using principal components. So now we can visualize the data in 2 dimensions. So when we are doing clustering, we are actually working with the large dimensional data. So if you look at this data set, I have 2 examples here. The first one is the Ferretti gut data set, and the second one is the Ferretti oral data set. So on the image on the left, I have the true groups, and on the image on the right, I have the estimated group for both of those data sets. For Ferretti oral data sets, our approach was able to give perfect classification. So it was basically able to recognize, just looking at microbiome profile, which measurements were from adult and which measurements were from individual. In the Ferretti gut microbiome data set, we actually misclassified 2 observations, and we wanted to see which were the observations that were misclassified. And I'm showing the 2 misclassified observation here with the circles here. And if you look at the 2 misclassified observations, so here it's actually kind of mixed with other ones. So if you look at the red dot here, this was an adult, but their microbiome profile is actually more similar to an infant. Similarly, here, this is actually an infant, but the infant's microbiome profile is much more similar to an adult. So even though it actually had 2 misclassification, it actually is identifying observations that are similar. So now moving on, as I mentioned, we have had the opportunity to work on various different data types. So now we are working on developing integrative clustering platforms that can incorporate information from various different platforms together. So multi-view data sets are essentially data sets where measurements on the same set of individuals are collected from different sources and different platforms. So each different platform provides a unique, but it's providing us a partial view. So right now we are developing integrative clustering technique that can cluster these multi-view data sets, but that also gives us a global view of another underlying problem, but as well as gives us a platform-specific view of the problems as well. And again, these are just some of the examples of our ongoing work. More detailed information about my work is available on my website. And if you have any questions, please do feel free to send me an email at sanjina.dang@carleton.ca. Thank you.
Fantastic. Thank you so much. There is a question that's popped up. How computationally difficult is clustering? Back in the day, I was writing code for 24-bit to palletized colors using clustering to pick representative colors, which took a little while to do. But now you have cloud computing and more data. So what are some of the challenges and the difficulty in the problem?
So I think like a lot of these computational challenges also comes around with the kind of models that you're using as well. So for example, most of the work on microarray data relied on a normal distribution, which has closed form solutions. So everything is very easy. It could be run in minutes. But now a lot of my work now focuses on discrete data, which the multivariate extensions of. So when we are talking about multivariate extension, we are looking at more than one gene at a time. So the models that are typically used for this type of data is extremely computationally intensive to fit, which is why, to make things simpler, people would just assume that there is no correlation between variables and just keep using the univariate approaches. But right now, what we have actually found was we have found an approximation-based technique that allows us to model this data at a much faster speed, but also provides a really good approximation. So again, the larger the data set is, the more computational time it will take, of course. And then we are also trying to develop these models to be able to efficiently handle large data set, with where we might. So for example, in gene expression studies, you are measuring thousands of genes. When you're thinking about genome-wide association studies, you would be measuring millions of SNPs. So these data sets are huge. And then of course, that comes with some additional computational challenges, but we are trying to find efficient and more elegant solutions to be able to handle these type of problems.
Fair enough. Yeah, and it's just, yeah, that's amazing. Speaks to the interdisciplinary too. You're looking at biology, you have to work on the computations, the statistics. It's a fantastic combination. Great experience for students, I'm imagining, to work in this area. Thank you so much, Dr. Dang. And just last but not least, we do have Dr. Corinne Millard, who's going to take the stage now to talk to us about her work.
Thanks very much.
Thank you.
Right there. Okay, so I'm Corinne Millard. I'm an assistant professor in the Department of Geography and Environmental Studies, and I'm a remote sensing expert, and I specifically look at remote sensing of the environment. So today I'm going to talk about some of the opportunities we have using data science and also some of the challenges that we have, and sort of the thing that brings together the data scientists and geographers in remote sensing. So there's a huge opportunity for big data analysis in remote sensing. Right now, the Earth is being monitored daily by hundreds of Earth observation satellites. So these are satellites that are specifically looking at monitoring change on the surface and mapping the surface of the Earth. We have this massive archive of data that's continually increasing in size. But within this archive, we have data of all kinds of different types. So we have different spatial, temporal, and radiometric resolution. We also have different types of sensors. So we have passive sensors, which is what you guys probably think about when you think about remote sensing. What we see in Google Earth and Google Maps is reflectance from the sun, passively measured at a satellite. We can also measure parts of the electromagnetic spectrum that you can't see with your eyes, and that can tell us different information about the surface of the Earth. And then active sensors are actually sensors like the one you see on the screen, where we actually are sending energy towards the surface of the Earth, letting it interact with whatever object is on the ground, and then measuring what comes back. And what comes back tells us about what is happening on the ground. And so these sensors can give us different information. And then, as I said, we also have it in all of these different resolutions. So it can become a challenge to work with this data for long-term monitoring. But our job in remote sensing is to collect, process, and analyze all of this data. And what we actually want to do is take the data and turn it into information that's useful. So some of the common tasks that we use in remote sensing are image classification. So Dr. Dang's presentation a minute ago that talked about clustering was really, really a good setup for me, because we do image classification using both supervised and unsupervised techniques. Our goal is to create thematic maps, though, of the surface of the Earth. We can also do monitoring. So monitoring things like insect infestations or doing change detection. So maybe mapping illegal logging in a forest or something. We also can develop models to estimate biophysical parameters. So in this case, we would have measurements on the ground of how much biomass is at a specific location. And then we would develop a model based on our reflectance or our backscatter from our satellite imagery to determine how much biomass is in a larger area and extrapolate those models outwards. This is really important because it shows the importance of measuring things on the ground as well to our work. So I'll give a couple of examples of some projects that I work on in remote sensing. The first is general land cover mapping. The example I'm going to show you is peatland mapping, which is my specialty within land cover mapping. So this is actually an image just from Google Maps. And this is Alfred Bog, which is a peatland that's east of here, halfway between Ottawa and Montreal. And what I wanted to do was create a thematic map of the different peatland classes that are in this area. So how would we actually go about doing that? Going from this image to, say, something like this, which is a land cover map of the different peatland classes. Well, to do that, we use machine learning. We use deep learning, we use AI, we use clustering techniques. So we're using all of these tools that are developed in data science and applying them to our specific problems. One of the big challenges is getting that data on the ground. So you can see these are my training data samples. They are spatially located throughout my peatland classes, and oftentimes they are temporally specific or, for example, location specific. I couldn't transfer this model necessarily to another peatland because the conditions are slightly different. So that's one of the big challenges that we deal with in remote sensing is collecting all of this data to have a meaningful sample that can represent a large subset of our different classes across space and time. In the past, we focused on using single images. So you can see this is my curve of vegetation over time. So in the summer, we have low vegetation. In the winter, in the winter, we have low vegetation. In the summer, we have very, very high vegetation biomass, and then it decreases again. We also have rain events, different lighting conditions, all sorts of things happen throughout the year that change. And because of computing power and processing and analytics and things like that, we used to just choose a single image and create a land use classification. But how do we know that that image is the best image? Well, it's really difficult to know. Sometimes you just choose an image based on availability. But recently, in say the last 5 years, we started using multiple images throughout the growing season. And so in this case, we've just triplicated our data. But we're collecting more of the growing season and understanding what happens in these different classes over time, because you might be able to separate a specific class, 2 specific classes in spring, but then another 2 classes in summer. So we try to collect images throughout the growing season, but picking those images is quite challenging. So recently, what we've started doing in remote sensing, due to the availability of all of this data and the ability to process it now, is using time series to improve our classification results. So grabbing images throughout the entire season, processing them, extracting relevant training data, and then producing a land cover map that way. And that leads to significantly improved results. Moving forward, we can actually use the entire time series, not just a single growing season. So in this example, I'm looking at a suburb part of Stittsville. So this is in 2018, where the Stittsville, or the suburb is completely developed and people have moved in or are living in the area. But if you look back in 2004, this was actually a forest. And so we can use the temporal time series signatures of these locations to look at when change occurred and then characterize other areas that have these same classes at different spaces, points in space and time. So if you look at the point B here, this is a wetland. It was a wetland in 2004 and it remained a wetland in 2018. The difference in color here is just due to the differences in seasons between these images. So no real changes have happened between 2004 and 2018. The bottom graph here shows the temporal signature of that wetland over time. The yellow or the orange line is the modeled signature. And you can see that there's been no interruptions. It's a wetland that has stayed a wetland. However, if you look at point A here, this is the forest that changed into a suburb. And we can see that with our model, we were able to model this temporal signature up to about 2005. Then in 2005, we had a massive swing, change in temporal signature completely. And then in 2009, it changed again. And so what that relates to, if you look at the imagery and actually determine what was going on at these periods of time, was that we had forest up to about 2005. Then there was a period of construction. And then the suburb basically was moved into, people planted trees and gardens and lawns and stuff. And now it has resumed this new temporal signature. So we can use this information, these temporal signatures, and combine that with machine learning, AI, to extrapolate these time series classifications to other locations and produce a, region-wide, nationwide, or even a global map, depending on, how much computing power we have. The second project I wanted to talk about was wildfire susceptibility prediction. Again, this is specifically in peatlands. This picture here is a pristine peatland in Northern Ontario. You can see it is really wet. There's a pond there. There's lots of small shrubby black spruce trees. And we don't generally think about these types of landscapes catching on fire. However, the picture on the right you can see is a burned peatland. And this was a severe fire. This peatland burned because it dried out, essentially. So it was no longer wet. The problem here is that with climate change, we have changes in precipitation regimes, and we actually aren't able to monitor these peatlands throughout the entire boreal forest. So we want to use remote sensing to try to monitor the surface conditions. And so we're using Sentinel-1, which is an active remote sensing satellite. At the top here, you can see the raw data, and this is information about the peatland over time. You can see it's pretty messy. There's lots of information there, but lots of data there, but not a lot of information that we can pull out of it. The yellow line here is the date that a fire occurred in this peatland. And so what I've done is we've used seasonal detrending analysis to extract the seasonal component and the noise, and that leaves this detrended signal. And what we can see here is that at the time of fire, this peatland was in drought. So the backscatter from Sentinel-1 was significantly reduced. And what's really interesting is that we see this in peatlands all across the country that burned. And so we're using this sort of modeling technique to try to extrapolate that and predict when a peatland would be likely to burn. This is preliminary results, but basically what we're looking at here is what the probability of any given peatland in Canada would burn in June 2021. So we took the Sentinel-1 data and said, based on these values, how likely is it these peatlands would burn? And you can see that there are some relatively high values, but there's nothing extremely high. As I said, these are preliminary results, but the idea is to give this type of information to fire management agencies so that they can monitor and say when a peatland might potentially burn. A peatland will only burn if there's an ignition source, so they have to watch basically to be able to protect these peatlands. So throughout my talk, I've talked about all of the imagery we have available, and you saw examples of hundreds of points of data and time series analysis. And so we're really imagery rich, but we're still relatively information poor. We have a massive lack of labeled data. We don't have enough people on the ground, like I'm in these 3 pictures here where I'm collecting field data, collecting measurements about land use classes or about soil moisture or biomass conditions. And that's because most of the sites that we deal with are remote and they're difficult to access, or at least a lot of them are. We often also have global or national study areas. Because we're able to process this imagery over large scales, we aim for these really big projects, but we often don't have enough field data to back up the information that we need in order to create the answers to the problems at these large scales. And then the final problem is just that we do need to have expertise in all of these different areas. So we need to know how to use the data science and computational science tools, but we also need to have an understanding of the physical geography in these areas and the biology of what's happening at the surface, and understand these constantly changing conditions and these interacting conditions that are going on in peatlands or in whatever area we're studying. So we have these special challenges. I'm sure every discipline has these special challenges, but for me, this part here, the collection of the data and the really understanding of the how this information is going to play into the data science is really, really interesting and really exciting and challenging. So I think it's a really great time to be a geographer where we have all of this data available to us, but we need to go do the fun stuff, which is go out and collect all of this field data. That was my talk. I just wanted to thank my lab for helping me put this together. If anybody has any questions, feel free to send me an email or find me at Carleton.
Amazing. Thank you so much. I know we started late, so I hope that you'll indulge us, a minute if there's a question or two that pops up in the chat for everyone who's listening and getting ready for lunch. I did have a quick question in terms of, you mentioned the challenge and the interdisciplinarity. It came through loud and clear, which is so exciting. Training students in this area, what sort of background are you looking for? What's helpful? What's missing? What's needed?
Yeah, really good question. So most of my grad students are geography students who are actually, the master's students are in the data science concentration, but they need to have strong computational skills, as in, knowing statistics and things like that, and programming skills. That said, I've hired a couple of undergraduate assistants who have a dual background in geography and computer science, and they're really, really helping us significantly bridge those two gaps and to let the geographers just get over those little hurdles that they have sometimes.
Amazing. Makes sense. Okay, great. Well, listen, everyone had a chance to share their research with us today, and they've given you an opportunity, ways to contact them if you have other questions. I think it was clear from what we heard today, the opportunities for collaboration are there. These are really exciting research areas that I think impact a lot of people who are in the audience. So please do reach out. I think we're out of time for any more questions. But with that, I just want to thank everybody for participating in this panel. Thank you to our panelists. Thank you to everyone who was listening. And I want to encourage everybody actually to go off to the poster session during your lunchtime to interact with people there. And hopefully you'll have a chance to run into our panelists virtually on the platform today and ask them any more questions that you may have. So with that, thank you so much. Enjoy the rest of the day. And thanks again for participating.